Hey guys, welcome to URC, also known as US Railway Company. Today we will discuss how we will use replication of prokaryotes to expand our railway system. First, let's talk about the different components and the purpose of prokaryote replication. The purpose is to produce two identical copies of railway DNA molecules, which is important for railway division. Prokaryotes are organisms that lack a nucleus and have a single circular chromosome that contains DNA and its helical structure. Our DNA railways are composed of nitrogenous bases that connect via hydrogen bonds. It also contains a deoxyribose sugar and finally the phosphate group. DNA chain is composed of a bunch of phosphate groups connected together. Here is a quick rolling circle example that visually explains the process of DNA replication. Now, once we have a railway strand, we have a certain section called the origin of replication, which initiates the process of replication. We send out helicase 5000 to the replication fork to break down hydrogen bonds between the two railways. To prevent supercoiling, we have gyrase 5000, which goes after the helicase and after the replication fork to prevent any supercoiling or to resolve any supercoiling. Now, once we've solved the supercoiling, we have to worry about the strands behind the replication fork, and that's why we need single-stranded proteins to stabilize them and to prevent any hairpin complications. Our workers start work from the 5' to 3' for maximum efficiency when dealing with the leading strand, which is 3' to 5' towards the replication fork. Issues arise when they get to the lagging strand, which is 5' to 3' towards the replication fork. They become exhausted and have to work in fragmented pieces as seen here. Now, we send our experts from the primase synthesis factory, which lay down RNA primers to match up with the DNA strand. Once RNA primers are down, this triggers the development of Okazaki fragments by DNA polymerase III, which only adds nucleotides to the 3' OH end of a primer. Following the Okazaki fragments, we send DNA polymerase I, which comes in and checks on the work that we have done. It ensures that no errors have occurred and also initiates exonuclease activity, which removes RNA primers and replaces them with DNA nucleotides. As you can see here, you'll see the RNA primers go from green to brown. The gaps between the Okazaki fragments are then filled by DNA polymerase I by adding DNTPs, and the fragments are sealed by DNA ligase. DNA ligase helps with the formation of the backbone of the DNA and the phosphodiester bonds. Now, you end up with two identical DNA tracks which will carry information and will then be used in the future as templates for the next DNA replication. Now, what happens if DNA polymerase I loses its ability to properly remove RNA primers or sealing of fragments? Well, the segments of DNA affected will be deemed non-functional until another batch of properly functioning DNA polymerase I comes in and removes the RNA primers and seals the gaps between the fragments. Thank you.